Good afternoon. My name is Salam Temitoko Jolakwe, and I'll be facilitating our workshop this afternoon on the identification of microbes. And I'm sure that Dr. Taiwo has already introduced us to some concepts and bioinformatic tools that is needed for which will be used as a total package and applied in the identification of microbes this afternoon. Now, I will be starting this presentation with a question. Why identify microbes? Well, about 1% of bacteria has been studied and discovered, and bacteria constitute a large proportion of microbes that we are generally interested in. Now, microbes are capable of so many abilities, and some of these their abilities that gives us concern is that some of them cause diseases that harm human health. However, some are beneficial as they are important components of our food chain, and some are even applied in food processing, just to mention a few of their beneficial uses. Therefore, it is necessary for us to have a proper understanding of what microbes we are actually working on, what they can do, and the first step is to actually identify them. Now, how do we identify bacteria, which is going to be the main focus of my discussion and training today? There are three major methods that are employed. First is the morphological identification method. Then it's split into two, where first you actually look at the colonial morphology of the bacteria based on the growth pattern on plates. Now, for the organism that we're interested in here, which is Bacillus megaterium, we can see that it grows as a single, creamish colony with round edges on plates. This is just some of the characteristics that we're interested in when describing the colonial morphology of a bacteria. Now, for cellular morphology, this is usually obtained from the result of Gram staining. Now, Gram's reaction gives information on the shape, the size, and the accumulation of peptidoglycan in the cell walls of the bacteria, which makes them either positive or negative. A purple coloration makes it positive. A pinkish coloration makes it Gram negative. Now, for the plates of figure on the left, we have a Gram positive bacteria because it is purple. It is rod shaped, and you can see that the rods are fairly large. Now, to the image on the right, it's Gram negative because it's pinkish. And then we can see that the colonies are really small, the cells are really small, and then we can see that these are cocobacillus. These are kind of like cocoid rods. Now, how do we identify bacteria? We continue by biochemical characterization. Now, biochemical characterization relies on the results of products of enzyme activity that are detected in the bacteria. Then this is often done with the API kits, the Analytical Profile Index kits, which is sold by Biometrics. Now, for our specific focus this afternoon, we will be interested in molecular characterization of bacteria, because this is the most reliable method as it relies on the information in the DNA of the organism. Now, for bacteria specifically, we're interested in the 16S RNA region because it is highly polymorphic, and this is the region that is amplified for identification of bacteria. Now, to focus on molecular characterization on bacteria, there are two large procedures, basic procedures. There's the wet laboratory procedure and the dry laboratory procedure. For the wet laboratory procedure, it involves DNA isolation. This is done from fresh broth culture of the isolated colonies of the organism of interest, which should be in about 16 to 18 hours of growth. Then followed by PCR amplification of 16S RNA region using appropriate generic primers. And then agarose gel electrophoresis for estimation of the amplicon size. This is followed by purification and sequencing of PCR products. Now, this entire wet lab procedure can be done by some companies which are involved in identification or sequencing of 16S RNA. All you need to do is give them a plates with isolated colonies of your bacteria of interest, and they run it all the way to the sequencing. Then sometimes also They also accept your purified PCR product, meaning that you have to do the DNA isolation, the PCR and gel electrophoresis, and then you send the PCR product to them. Of course, this has implication on the budget of your laboratory. Now to the dry laboratory procedure. This involves the application of bioinformatic tools, and this is stated as sequence editing, assembly of contigs, sequence homology, sequence alignment, phylogenetic analysis. Now, if you have sent your samples or your organism to a company, usually the sequence editing and the assembly of contigs is done already. Then you just have to proceed from step 3 to step 5. Now, when the company sends your assembled and edited sequences, especially for a 16S RNA, it usually comes in a format that we call the FASTA format. And this is the FASTA format sequence of the 16S RNA for organism with code SF4, which has 1,492 base pairs of nucleotides in this region that was sequenced. Now, to sequence homology. The most common tool that is used is the NCBI Blast. I'm sure we are familiar with NCBI right now. So now this is what our NCBI homepage looks like. And then we are specifically interested in the Blast as indicated by the red arrow. Now when you click on Blast, it takes you to this page where it shows you the basic local alignment search tool and all the other mixtures of things you can do. Now we are specifically interested in nucleotide blast because our 16S RNA is a nucleotide sequence, and we are interested in searching for homology to nucleotide sequences in the NCBI database. Now, when you click on that, it takes you to the NCBI Blast settings. You paste your edited and assembled sequences here in FASTA format, and then we change the selections as indicated in the arrow. We'll see that in the next slide. Now, we change on the database that we are interested in to 16S ribosomal RNA sequences because that is what we have for bacteria. And then we also select Bacillus as the organism of choice because this organism that we are using for our hands-on practical today has been identified morphologically and biochemically to be Bacillus previously. Now, so we also select our program and we optimize for highly similar sequences, which is Megablast, because we are interested in identifying organisms with the closest homology to our unknown sequence, and then you simply click on Blast. Then our submission is in process. And when your Blast result returns, it returns in a new page. Then you simply scroll down to descriptions because that is where our important information is. Now, the first organism on the list here is the one with the closest homology to our unknown 16S RNA sequence. And here we see that it has 99% homology to Bacillus megatherium strain ATCC 14581. Therefore, our organism is a new strain of Bacillus. Now, in addition to the result, we see that we have a list of other bacteria that our strain shares common homology with. So for further analysis of sequence alignment and phylogenetic analysis, we need to download at least 10 of these sequences in FASTA format to a Microsoft Word document for further processing. Now, this is how you do it. You click on the colon with accession number. You can see the red arrow. Then you click on the links of the accession number, then for the first 10 at least. So when you click on this link here, it directs you to another page, a new tab on your browser, and then you can see the FASTA format, FASTA link here. You click on that, and then it takes you to this page, and all you need to do is simply select and then you copy. So you do this for at least the first 10 sequences, and then you paste this in Microsoft Word. So you paste it in the Microsoft Word like you can see that I've opened, and of course you'll be adding the sequences to your own organism of interest, which makes 11 sequences that you'll be using. So right now we'll proceed to the hands-on of this sequence homology, and then we'll come back later to continue with sequence alignment and phylogenetic analysis. Thank you very much.